Um, Good afternoon, everyone. I'm Aditya Ganesh. Today, my PGP topic for presentation would be embryology of eye. The embryology of eye is granted and slightly complicated topic to explain, and it can get convoluted at times when we initially begin it. Yes, and the embryology of eye can get a bit complicated if we do not have a basic understanding or a basic idea of how the basic structures originate or the basic terminology of embryology. So this presentation is mainly aimed at making sure that we all can understand the basic steps which are there in embryology. So the basic goals and objectives of this presentation would include obviously getting the basic idea of eye development, understanding the terminology, and the clinical significance. The reason I've highlighted the clinical significance is basically when we tend to read embryology, we try to remember embryology topics in terms of weeks, which week what happens, which day what happens. And that can get confusing at times. But in this presentation, I'll be focusing more on the clinical aspect if an embryological event does not happen accordingly, or if an embryological event does not happen completely or it gets interrupted in between, what is the clinical manifestation of that? We will be having a clear idea if we can understand the basics of embryology. That's the reason I highlighted the clinical significance. Now, whenever we read about topics, we may hear about colobomas, the word coloboma, and we may also question like how do colobomas form. And sometimes whenever we read about retinal detachment, we always see that the detachment occurs between two certain layers. I'll be mentioning what layers, and I'll be mentioning why does the retinal detachment occur as well. And basically, why is exactly the optic nerve susceptible to neuritis? And basically, why is not any other nerve susceptible to neuritis? Why optic nerve is susceptible to neuritis? We'll be learning if we know the embryology. Now, the moment we open our embryology textbooks, we'll be facing these tables, the derivatives of ectoderm, mesoderm, endoderm, the day, the week, etc. It can get really confusing, and within 2 minutes, our attention span goes out of the spanner. And if you try to look at the pictures as well, these are pretty much complicated with lots of complicated terms, lots of complicated labeling, multiple layers in the eye, and then it can become a bit overwhelming as well. And if you try to simplify and look at the flow charts, this is even more confusing because you will definitely get lost within the first few seconds, and you'll be developing ocular saccade just by looking at the flow charts. So the objective, as I mentioned before, is just to get the basic idea of embryology. So let's go with the basics. We might have seen this picture multiple times when we are starting with embryology of any topic. We know that we know that the zygote, we know the morula, we know the blastocyst, the multiple stages after which the fertilization happens. We know that there's something known as gastrulation as well. We'll be coming to them in a short while. But basically, I would just like to throw your attention to this small part over here, this last structure over here. Now, if we are to have a closer look at this, we see that there is a small embryo which has multiple structures, multiple folds as well. And now as the embryo keeps on developing as the weeks progress, it slowly starts resembling an embryo which is of a human origin. Again, as the weeks progress, as you can see, the limb buds develop as well as the cranial neurological development as well. But we are focused mainly on the eye for today's lecture. And if we are to look at what step is basically more significant, we have to focus on these last two steps. And if we are to take a cross section of this area, we'll be ending up with this complicated structure. Now, at first glance, you might be seeing a lot of multiple terms such as notochord, neural tube, mesoderm, neural crest, all of them which sound very similar, very confusing. So let's try to simplify it as much as possible now. So basically, we know that the eye has gastrulation is involved like with three layers. We have the ectoderm, we have the mesoderm, and we have the endoderm. Basically 3 layers. And quite thankfully, to make our embryology much simpler, the endoderm does not play much role in the development. So the ectoderm and mesoderm are the major two components of this. Now, there are a sequence of steps which happen which lead to this formation. There you can see that I'm just highlighted the ectoderm, and we can see the ectoderm. There happens to be a lot of steps which are happening to the ectoderm, neuroectoderm, surface ectoderm. It kind of invaginates inwards, etc. And at first glance, it may look a bit complicated. Let's try to look at it step by step. So here I'll be just drawing the ectoderm. So this ectoderm, just the ectodermal layer. Now this ectoderm kind of differentiates into two parts. Let me draw it again. Ectoderm differentiates into two parts. So this part is something known as the surface ectoderm, and this part is known as the neuroectoderm. Now there is a significant event which happens with the neuroectoderm, is that when the surface ectoderm, there happens to be an invagination inside. It kind of folds inside, and then which part folds? The neuroectodermal part. The neuroectodermal part folds inside, whereas the surface ectoderm still lays outside. And then as the fold continues, it kind of separates from the top portion and forms a tube. This is still a neuroectodermal derivative. This is surface ectoderm. And I would just like to show a small demo of what exactly happens. If I can pause the video for a second, and I can show you a demo of what exactly is happening. Yeah, so basically, we might see ectoderm, mesoderm, endoderm as three layers. I would just like to represent these three layers in form of a book. You can easily imagine that the ectoderm is over here, the mesoderm is in the middle layer, the endoderm is in the lower layer. Now, let's simplify it further. We are now just looking at the topmost layer, the ectoderm. So if I keep this aside and just take a paper for more simpler representation of the ectoderm. As you can see, I have highlighted two different parts of the paper. One, as you can see, is there's something differentiated in between. This, as I mentioned earlier, is something known as the neuroectoderm, and this is the surface ectoderm. Now what happens? We all know that, as I have drawn earlier, the neuroectoderm kind of invaginates inwards, and you can see there's a small V fold which forms with the neuroectoderm. And then, as you can see, the neuroectoderm kind of fuses together. It forms a tube. The neuroectoderm fuses together and forms a tube. As you can see, a tube which runs all along. This is a neural tube formed by the neuroectoderm. And this is the surface ectoderm. If you look at the top portion, the surface ectoderm is like that. But if you look at the bottom portion, the neuroectoderm is like a tube. And now one more thing I would just like to show is that when it is folding, it actually has two folds which form at the either side. If it is visible in the video, it's not a straight line. There are two folds which happen when the neuroectoderm kind of invaginates inwards. One fold over here, one fold over here. Now this is a significant step which happens. Let's keep that in mind. Let's keep that in mind and focus on this part as well. I have drawn something green over here. They are just near the neural tube. Above, you can see at both sides of the neural tube, there are like green structures, green stuff, which is hanging around. Now, I will not name that structure right now. Let's look at the terminology. I'll be explaining what, but just remember that there is some green cells which are hanging around in the neural tube. Now, as this is getting a bit tough to handle a big paper, let me take a smaller cross section. This is a neural tube. And you can, as I mentioned before, there are the two folds. Now, you might be wondering, the surface ectoderm will lie flat, but in all the diagrams which are given in our textbooks, we might be thinking that the surface ectoderm kind of does not play any other further role. But you have to remember development of embryology, development of the embryo is a three-dimensional structure. This surface ectoderm actually is bent, is kind of semicircular, attached to the endoderm below. So basically, the embryo is like this. So we can see basically you can see the neural tube which has invaginated. This is the neuroectoderm derivative inside the surface ectoderm. Now what happens is, as you can observe, there are two folds. There is a significant event which happens at these two folds, and there happens to be an prolongation of this two growth to meet the surface ectoderm outside. Now let me make it more clear by drawing it. Let me just start my screen share again. Yes, so basically this is the last picture we've seen right now. We saw the surface ectoderm, which is kind of like folding this side, and we know that this is a neuroectodermal derivative. Basically, this is nothing but the neural tube. This is a neural tube. Now, what happens is there happens to be a significant event which is very important when during the development of eye. What happens is that from these two ends, there happens to be a development of a stalk-like structure. As you can see in the diagram, the two ends the stalk structures develop, and then a small vesicle forms at either end. As you can see, a small vesicular structure forms at either end. Now, this is a meeting, or I would like to term it as a meet and greet between the neuroectodermal component and the surface ectodermal component. Now, let's just focus on this part, just this part. Let's go on to the next slide. As you can see, this is the optic vesicle which is coming from the neuroectodermal derivative, and this is the surface ectoderm which is lying near this. And as it just keeps on going, progressing forward, there happens to be once the neuroectoderm and surface ectoderm kind of come in contact with each other, there happens to be a significant event which happens. What happens is the surface ectoderm, which is lying over here, gets slightly thickened, and there are a lot of changes which happen, which form the lens placode and the lens pit. So the further events, which I will show it as such, and as you can see, the lens pit kind of invaginates inward, and the neuroectoderm kind of encapsulates it as well. And then finally, we find the lens vesicle, and we find the optic cup. So based on the small demo which I had given you and the diagram which I'm showing you, perhaps it may be slightly more clear of how the actual events are taking place and how the structures are being derived. So this component is the optic stalk, this component is the optic cup, and this is the surface ectoderm, and this will obviously form our future lens, which I will be explaining further as well. Now, again, before we jump too deep into the embryology, it is essential that we know the terminology. As I mentioned, neural crest cells, mesoderm, mesenchyme, neuroectoderm, all of them sound very similar, can get very confusing. If I can spare just a minute to explain the terminology, it will be much clearer to understand the embryology. We know the basic three layers: the ectoderm, mesoderm, and the endoderm. Now, we already have seen that the surface ectoderm kind of differentiates into surface ectoderm and neuroectoderm, and further progressing to form the neuroectoderm kind of invaginates downwards and forms a neural tube. Now, remember, I told that there are some green structures which are hanging around. This is called the neural crest cells. Please remember, this green slime is going to be accompanying us till the end of the lecture. This is the neural crest cells, and we have the mesoderm on either side. Endoderm is, we do not bother with endoderm with this lecture. Now, let's focus on this surface ectoderm. Let's focus on this. So we have the neural crest cells, the green slime, and we have the mesoderm. So in order to make it less confusing, let's not split it into neural crest cells and mesodermal derivatives. Both of them together is called the mesenchyme. And I will give you a small limerick. Mesenchyme is the slime. Please remember, this is our slime which we are going to be handling throughout the lecture. Just remember this point for now, so that as we progress throughout the presentation, it will become much easier. Neural crest cells, the neural crest cells over here, along with the mesoderm, form together are the components of mesenchyme. Now, I put this picture just for representational purposes. As you can see, a racetrack, lots of players over there in the race. Now, if you want to really understand how a race occurs, if you just look at the game, you just see the result, but you do not know what components and how the race exactly happens. So embryology is not like step by step. That means if the retina develops, only then the lens will develop, only if the lens develops, only the cornea. No, they all happen kind of simultaneously. So it is very important for us to visualize each and every component separately and then combine them together instead of reading everything simultaneously, like what we were doing in the table earlier. If we do that, we are going to get confused and get lost in between. So let's understand each and every player's characteristic by looking at each and every component. So let's have a general overview. Now this picture will be coming up a lot of times in this presentation. We can see week 3, week 5, week 7, week 8. There are a lot of stuff which is happening over here. Just to make things simpler for us, let's remove the labeling and just let's focus at the pictures. Look at, we might recognize this and we might recognize this. We don't know yet what these two pictures represent. We know that the surface, this is the surface ectoderm, sorry, yeah, this is the surface ectoderm, this is the neurectoderm, which kind of invaginates inwards, and this which comes like this neurectoderm kind of comes like a optic stalk. The surface ectoderm in the side kind of invaginates. So we know this two, we don't know either these three. Now keep in mind, in this picture there is again our friendly slime mesenchyme hanging around everywhere. Just keep that in mind right for now. So let's look at each individual player of the race. We have our optic stalk and cup. So let me just represent this optic stalk and cup. Optic stalk, this is the optic stalk. We have the optic cup. We have the lens vesicle, which kind of invaginated earlier from the surface ectoderm, if you remember. And we have the vasculature. I will explain how the vasculature also plays an important role in the development, in the steps of embryology, and what happens if something goes wrong. And of course, we have the development of retina, which occurs by the involvement of these two layers, and the adnexa. Adnexa basically not only involves the eyelids, but also the muscles which supply the surrounding structure. So these are the basic components of embryology. If we just go through each and everything of this component, understand, and then combine them in the end, it will become much simpler for us. So let's just focus on optic stalk and cup. When does it happen? Not very important, but we just need to remember it happens in third week. Where does it happen? Now, something known as prosencephalon. In primitive brain is kind of divided into three components, which I'll be explaining further, but for now it's prosencephalon. Why does it happen? Optic stalk and optic cup are responsible for the formation of these four: the neural layer, the retinosa pigment epithelium, iris, and ciliary body. Both of these constitute actually the retina as well. Now, you might be thinking, what is prosencephalon and diencephalon? If we have to take a look at the picture, as I was shown earlier in the 3D model, there happens to be a tube which kind of runs down the embryo. So this is nothing but this tube. This is the prosencephalon. The front part is prosencephalon, midbrain is mesencephalon, the hindbrain is rodencephalon. The prosencephalon kind of differentiates into two parts: the telen and diencephalon. And if you remember our two friendly stalks and structures which are kind of originating from the tube, yeah, this is actually the point of origin from this. So basically, you don't need to kind of remember the telencephalon and diencephalon. Just remember that optic stalk and cup kind of form from the frontmost part of the tube, which is the prosencephalon. And again, we have a very famous picture from Wolff's anatomy. We can see our wall of prosencephalon. From the wall of prosencephalon, there appears to be a stalk which kind of develops in the front. And we have the stalk kind of forms an optic cup. Now look closer, you can see there are two layers. One layer over here, another layer over here. We have the optic stalk and optic cup. Optic cup constitutes of two layers. Now these two layers are very important in the formation of retina. Why? The two layers, the front layer forms the nervous layer, which is the neurosensitive layer, and the behind is the pigment layer, which is forming the retinosa pigment epithelium, which is located behind. Now this kind of gives us a clear idea that there happens to be a small cleft between these two layers. Now just because you understood the embryology, now you'll remember that whenever there happens to be some kind of a retinal detachment between the pigment layer and the nervous layer, it happens between these two layers, the neural epithelium and the pigment layer, because of the embryological development. So any fluid or any way it kind of detaches, it usually kind of tends to detach from these two layers. And this is the front part, and and this is the behind, and there are also certain events which are happening in the front. I will tell you. Now, look, as you can see, again, let me draw the optic stalk. We have the optic cup. We know that these two layers kind of form the retina, and we know this is the lens placode. Now, what happens over here, we have to focus in the front as well. This kind of invaginates inwards like this. And we have the surface ectoderm as well over here. Now, you can have, this is a very rudimentary and a very bad diagram, but I'm pretty sure you can identify what this structure is, which will form the future iris. And this is the significant events which happen within optic stalk and cup. Front is the iris, back is the retina. Now, you might be thinking that this portion over here is fully cylindrical, but actually it's not. During the initial phases of development, We might have a misconception thinking that this is kind of cylindrical, but it is not. It is kind of semi cylindrical like this. Sorry, I'm very bad at drawing diagrams, but this is just a representation. As you can see, the optic stalk is kind of hollow from below. It is not fused. There happens to be a cleft below it. Now, why is this significant? I will mention why it is significant. Again, this is a line diagram of the optic stalk, optic cup. And as you can see that there happens to be a small fissure below. And it is not completely fused bottom. This is known as the choroid fissure. So if we are to take a choroid fissure and take a cross section like this, and then we can see the hyaloid vessels, the lens placoid, the layers of the retina. This is just a cross section of this part when we are taking it in this direction. And because the bottom part is kind of not complete, this allows the hyaloid vessels over here to kind of travel inwards into this cleft and supply the lens placoid. So this is the role of why there happens to be a choroid fissure. Again, if you take a cross section of this part, you'll get this, and we can see that it's not completely fused below. Now, what if the choroid fissure doesn't close by 7th week? The fissure closes by 7th week, basically. But what happens if it doesn't do so? And we end up with a very famous clinical condition known as the anterior coloboma and the posterior coloboma. If I can go back, you know that this part, two parts invaginate like this, forms the iris, and the two layers behind form the retina. Now, if this part is completely compromised in forming, we can see that the iris is not completely formed below. We find a keyhole defect in the inferior part because it is not completely fused in the bottom. And if you have to look, so the iris, there is a defect in the forming. That's known as anterior coloboma. Let's go back and we see the retina over here. If the retina behind does not fuse as well, we find the posterior coloboma, which is pretty well represented in this picture. This is the retinochoroidal coloboma. You can also, there's also another coloboma, which is the optic disc coloboma as well. Now, I would like to show a picture which we have seen in one of our calls. Is an patient having, we got called for an SICU, and we happened to find this patient also has an coloboma, anterior coloboma of the iris. We did not do a fundus, but we know that this patient is having an anterior coloboma. And if we are to palpate the lower orbit as well, we found that even the orbital margin is kind of deficient between. So basically, one part of the face did not completely fuse yet. So we can find the deficiency even in the margin of the eyeball as well as the iris. Now let's go back to our old slide, which we told we saw it multiple times. Let's focus on these two parts. We have these two pictures. We know our friendly neuroectoderm, our surface ectoderm, which kind of invaginates inwards. Now let's take a cross section of this part again. So we end up with this picture. We know that it is not completely fused yet. Now what happens after it fuses? Now you have to remember the optic stalk, which kind of comes from behind to front. Now this optic stalk is responsible for harboring all the optic nerve fibers. If I am to again go to the next picture, as you can see, we have optic nerve axons which are actually traveling inside this stalk. This space kind of differentiates into forming the optic axons. Just imagine a PVC pipe and there are a bunch of wires in it. That's basically the optic stalk. And imagine the wires inside are separated by rubber bands. These rubber bands are nothing but the septa which are formed by the astrocytes. Now, remember, I have been mentioning a lot of times that there are green stuff which kind of hangs around our mesenchyme, the slime mesenchyme. You know that from when the stalk is kind of developing in front and going and forming the cup, our green stuff also goes along with it. Now, because we have taken a cross section over here, we can see our mesenchyme kind of hanging around the optic stalk as well. This mesenchyme condenses together to form the sheaths of the optic stalk. Now, why am I mentioning that this is an optic stalk? Why am I mentioning this is condensing? You might be, if you ever wondered the term optic nerve, basically, I feel it's a misnomer. Optic nerve is technically not a nerve, it's just an extension of the brain. And we might be confusing optic nerve with basically other nerves which are present in our body, other nerves which are not components of the central nervous system. If you think back, we know that all the peripheral nerves are Schwann's, the Schwann sheath or the covering of the nerves are formed by Schwann cells. But The central nervous system, the coverings of the central nervous system are not formed by the Schwann cells. It's actually formed by the oligodendrocytes. Now, why am I mentioning oligodendrocytes? Why am I mentioning that this is a part of the central nervous system? You have to remember oligodendrocytes form the myelin sheath, but not the Schwann cells of the optic nerve. Why am I mentioning this? Because this becomes important in the condition of optic neuritis. Optic neuritis is kind of very common in conditions of multiple sclerosis. If we remember in medicine, multiple sclerosis happens when there is an autoimmune response against oligodendrocytes. That's the reason most of the involvement of the brain, different place and different time happens because the oligodendrocytes are getting affected. So basically, the central nervous system, the myelin sheaths are getting affected. Once the myelin sheaths are exposed, it gets prone to inflammation. And that's the basic main reason why optic nerve is more susceptible to, optic neuritis, what you call neuritis per se, because of it's prone to inflammation because the oligodendrocytes are forming the coverings. And you might also see that I have mentioned other two conditions such as myelinated fibers and optic nerve hypoplasia. Why it is happening? Again, we have our optic stalk and optic cup. You have to remember the nerves grow inward, whereas the myelination occurs in the opposite direction. So the myelin sheath kind of condenses over here, condenses over here. Technically, it should stop at one point, which is known as the lamina fibrosa. It should stop at one point. But if it doesn't happen to stop at one point and it kind of progresses on to the outer layers as well, we find that the myelination is happening. We can see on fundus examination, the myelin is kind of getting spread over here as well. This is the myelinated fiber condition. And if there happens to be less number of axons in the optic stalk, basically less number of wires in the PVC pipe, we find an optic nerve hypoplasia. Now, as I mentioned, we have the optic stalk, the optic cup. We know from the optic cup the iris forms, and in just a brief mention in that formation of the iris, the sphincters which are present in the iris are actually derived from these components as well. So all the sphincters are forming in from the iris when it kind of invaginates inwards and forms like an iris in front. Finally, we come to the favorite topic, the lens, most favorite topic. Again, let's go back to our line diagram. We know our optic vesicle stalk kind of invaginates inwards, forms a lens vesicle, optic curve, and a surface ectoderm. Now, I have just given a small representation or a line diagram of what's exactly happening. 1 second. Yeah. Now, imagine the lens plecote as a single row of cells. What happens is, I would like you to focus that there are two halves of here, anterior and posterior. Now imagine the posterior cells elongating from behind. These cells elongate and kind of fill this cavity, and let this elongation kind of proceed further as well. And it will completely occupy the cavity as well. And you'll be still thinking of what about this anterior half of this cells of the lens. What happens is they will not stay stagnant. They are also active. They are also kind of forming another capsule all around. So they kind of invaginate and then form multiple rows of cells all around the developing lens. So basically, this picture I have represented it as an animation. Now, one small thing I would like to mention, a small theoretical part. The nucleus over here, kind of because the cells are elongating from behind, the nucleus are still like this. So this is nothing but the lens bow. Basically theoretical part, but if you see from if you see microscopically the lens, the nuclei are arranged in such a way. Now what happens is that because of and one more thing which I have to mention is that the transparency of the lens fibers is happening due to the loss of cell organelles. All the organelles of this posterior cells kind of lose, and they lose all their organelles, and they begin to accumulate something known as crystallins. So these crystallins are important for giving the transparency, which is happening during the 7th week. Now let's focus on this lens again. You know that these are the rows of cells over here. Now let's imagine that these rows of cells are now kind of compressing together. Now the full growth happens. Now these rows compress. I call it the squeeze. All the rows are compressing around the posterior fibers and form a solid lens. Now during the squeeze, you might be seeing that there may be embryonic suture or Y-shaped suture because of the overall arrangement of the fibers. we might see a small Y-shaped suture, which is nothing. We can also see and appreciate it in slit lamp as well. These are the Y-shaped sutures around the embryonic nucleus. If you happen to have a clear media, we can sometimes visualize the embryonic Y-shaped sutures in certain patients as well. Now, at least by looking at the line diagram for like 100 times right now, we at least can understand what is this histological picture over here. We know this is the optic cup. We know this is the lens vesicle. And you might be wondering what are the small structures which are hanging around behind. Now, we have to realize that the lens is a bunch of cells which are kind of differentiating into multiple layers, and it becomes pretty hungry. The lens is kind of not suspended like in the medium. It needs supply. It needs blood vessels when it is in the growing stage. So that is important. That's the reason we need to look at the other picture. We'll come to the next two pictures. As you can see, we have the hyaloid vessels over here, and we can see there's a vessel which going from behind coming to the developing lens. This is important for our supply. So again, optic stop, optic cup, developing lens. The hyaloid vessels kind of come from bottom and then travel outside the cup and supply the lens. This is nothing but tunica vasculosa lentis. Tunica vasculosa lentis. So this is responsible for supplying the lens. This is a very good picture of how it is happening. Now, why am I mentioning that this is happening? And it is actually going through the vitreous. It is forming something known as a canal of Cloquet, which goes in the canal and supplies the lens. Now, what is the significance behind this? Before I explain the significance, again, let me just show a diagram. As you can see, the vessels are coming from behind, supplying it, forming a tunica vasculosa lentis. And in this part, we see embryology. We can see the primary vitreous, secondary vitreous, and tertiary vitreous as well. They are the three vitreuses which form when the blood vessels develop. Now, what happens? Why is this significant? Why? Because if any part of this vessel kind of stays behind, if these vessels do not tend to go away, and if they kind of stay as a remnant, each and every part of the vessel will form some pathology. Now we have the persistence of, if this kind of hyaloid vessels are kind of staying in this cavity, we form persistent fetal vasculature, persistent hyaloid artery. And if the glial components in the front are staying, we find Mittendorf dot and Bergmeister papilla. So these are the good clinical pictures of what is happening. Again, persistent fetal vasculature, we can see that the tunica vasculosa lentis is still persistent in this picture. So this is the clinical significance. Once we are done with lens, we are going to the retina. Again, we come back to the same picture: optic stalk, optic cup, two layers. Remember that this layer actually forms all our major part of the nervous epithelium. Now, how do you remember retina in an easy way? This is how I remember. I just draw 3 rows of axons. 3 rows of axons. Like this. And they are sandwiched between 2 limiting membranes. And we know our, let me draw something, rods and cones, representative picture of rods and cones. And most below, we know that there is a retinal pigment epithelium, which kind of lies below. So very, very, very basic representation of the retina. Now, if you remember this picture, remember the layers of retina as well. It is sandwiched between the internal limiting membrane. And one more thing, please remember, this is inside and this is outside. So we get the orientation, because we'll always lose orientation when you're drawing retina. We have the internal limiting membrane. We have the external limiting membrane. We have the outer nuclear layer. We have the inner nuclear layer. All the ganglion are arranged side by side. We call it as nucleus. So this is the reason we call it outer nuclear layer. Any axons which are arranged, which is plexiform. So this is known as outer. Oh, sorry again. Yeah, this is known as outer plexiform layer. Inner layer, inner plexiform layer. And finally, the outermost where the axons of the retinal nerve actually develop, which is the retinal nerve fiber layer. And this layer is known as the ganglion layer. Just 3 rows of axons sandwiched between 2 limiting membranes, rods and cones, RPE. And we obviously have our choroids below. That's it. If you remember this, the layers of retina, it's a very easy way to remember our layers of retina. Now comes a very slightly complicated picture. No need to remember each and every component. It's just to represent that in this neurological layers, multiple layers are originating from each and every layer. It is not highly essential for us to remember what layer forms what. Easy way to remember, because sometimes they may ask us like, what is the derivatives of Indian neuroblast? What are the derivatives of outer neuroblast? You can remember with mnemonic. I keep the call in game or rock on, whatever. Basically, you can use any kind of mnemonic to remember this. Now, again, let's go back to our whole picture. We are almost done with retina. Please remember, you can see, as you can still see, our green slime is hanging all around. Do not remember, do not forget that our green slime is always hanging around when the development of eye is happening. Why am I mentioning this in green slime? Because this mesenchyme, the slime, has the property to invade everywhere. We come almost to the last part of the lecture, the invasions. I term it as invasions because I did not yet discuss about a couple of components such as cornea. I did not discuss about extraocular muscles, adnexa, sclera, uvea. Why all these components can be easily explained due to the mesenchyme, the green slime, which invades everywhere into the already existing structures of optic corp, vesicle, and the ectoderm. Let's go to the invasions. Now we saw this picture. Let me again remind you of the terminology. Neuriferous cells plus mesoderm forms mesenchyme. Now you might be wondering. Let's pause for a while. Let's look back. We know that the ectoderm kind of forms the outer structures. We know the endoderm forms the lining of the most of the organs inside. Now what is there in between? It's obviously the mesoderm. The mesoderm technically forms something, the structures between the lining and the outer body surface. So what are the structures which lie between? Blood vessels, muscles. So if you have to remember this, according to general embryology, it's just a general thumb rule. It is not exactly, it is not a, what you call a law, but it's just like to remember, mesoderm generally involved in forming the vascular and muscle systems throughout the body. If you remember this one line, it becomes very easy to explain, like basically all the other structures which are kind of remaining in our embryological journey. Again, we come back to this picture. We already seen this. We already seen this. We have to see this again. The green slime is hanging all around, all around, all around. Mesenchyme has a peculiar property of getting into everywhere. It's just like going to the beach and the sand getting everywhere into your dress. So this is exactly how I remember mesenchyme. Now again, a small representational picture of what happens. You can see the green cells which are hanging around. Now what happens is something known as invasion. Whenever an invasion happens, everybody does. Whenever a war happens, it does not all happen at once. They invade like in waves, one wave, second wave, and third wave. Similarly, this is what happens when I mention invasion of waves. So first, basically, just focus on these cells. The cells kind of invaginate forward and form an endothelium. This row of cells is actually the corneal endothelium. Let me go back. So again, focus on this part and see how the cells are kind of going forward in the first wave. They're forming the corneal endothelium. The corneal epithelium is a surface ectoderm. Corneal endothelium is actually the traveling of the cells forward. And the second wave is not involved with corneal endothelium. It kind of goes inwards and fills up the iris and the uvea behind it. So basically the structure, the basic structure, the skeleton, it's a very wrong word to use, but I would mention the skeleton of the iris is actually formed from the optic cup, but the inside, the stuff inside the blood vessels, the choroidal system is actually due to the mesenchyme going inwards and invading everything. Again, remember, mesoderm forms our blood vessels and muscles. And finally, the third wave kind of forms the stroma of the cornea. So again, corneal epithelium, we have the stroma and the corneal endothelium. And we also, because of these waves, we also have the blood vessels which are kind of occupying into the iris and the uvea as well. But again, this is the same representation in the picture. As you may be wondering, this is just corneal epithelium, corneal endothelium. Now, why am I telling invasion? Just look at this massive invasion which is happening right now. The stroma cells are casually strolling in. You might be wondering, okay, fine, it's formed a small layer. And suddenly you end up with this big layer. This is the stroma because of the invasion of all the cells, all the mesenchymal cells, and for filling it up. Now, these cells are keratocytes. These keratocytes kind of lay the collagen in a lattice arrangement in this layer. This lattice arrangement is very important so that the light rays can travel and can get through the cornea very clearly. Now, what happens if there is some defect in this lattice arrangement, or the keratocytes are not invading correctly, or there happens to be a problem in this? What happens if the keratocytes of the stroma fail to produce the collagen in a lattice kind of arrangement? We end up something known as sclerocornea. Basically, sclerocornea is nothing but corneal scleralization. Whatever the cornea is supposed to be transparent, but it kind of appears like a sclera itself. So this is nothing but sclerocornea, very simple. The trabecular meshwork, if you have to look at the eye from gonio angle, if you have to look from this side, if you are looking from the eye like this and you're looking from the side, we see that basically you don't need to dwell too deep into how the trabecular meshwork works. You just need to realize that we have the iris over here, we have the ciliary body over here. I already mentioned that there is the endoderm which is formed from our green friends, and this green friends also kind of help to form the trabecular meshwork as well. That's all we need to remember. We do not need to know how exactly it invaginates and everything. Now, what happens if the canals don't form exactly? What happens if the meshwork doesn't form? We end up with congenital glaucoma. The drainage from the posterior to anterior gets blocked. The fluid kind of accumulates and forms congenital glaucoma. And we can also see a beautiful representation of the Habs striae, the multiple features of congenital glaucoma as well. Again, we are at the end, last 2-3 slides. We have our green friends again over here. We might have forgotten, I didn't mention about the sclera. Everybody has a very popular misconception that the sclera is formed from the outside. It is not. Our mesenchyme kind of, like how the mesenchyme condenses to form the dural sheets, our mesenchyme also condenses to form the sclera all around. So basically our sclera is formed because of the condensation of our mesenchyme all around. And then the muscles which are present in the eyelids, the mesenchyme also invades there and forms the muscle. It invades the iris, forms the blood vessels. It invades into the eyelids, forms muscles. So everywhere it kind of invades and forms most of the eye as well. One significant thing I wanted to mention when eyelid development is, we know that the eyelid actually initially separate, and then as the development progresses, they fuse. And then once again they will separate. So it is not always separate. They are separate, they fuse, and then they are separated from each other. Why is this fusion happening? This fusion is very important so that the glands which are present in our eyelids, the meibomian and etc., meibomian glands, Zeiss glands, will get some time to develop between when they are fused together. So this gland formation is very important. So again, let me just highlight the flow chart. We have the formation, we have the fusion, and we have the separation. Basic flow chart. Now, what happens if this process gets interrupted? What happens if the formation is gone? We develop with cryptophthalmos, because if the eyelid does not separate at the embryological development, it kind of stays like that, and the baby is born with something conditioned known as cryptophthalmos. And if they fail to fuse, if they're keeping on separated, they form something known as the lid coloboma. It is actually a kind of a misnomer, but let's remember it, because if they fail to fuse correctly, they form colobomas. And if they fail to separate, they're still kind of remnant, they're partially open. It's known as ankyloblepharon. So we are done. This is the last slide of the lecture, and I do not want to put up the tables, because again it is going to confuse us. So I found a really good video from the American Academy of Ophthalmologists, which I will play, which is just a 2-minute video of how the embryological development is happening. So let me pause and then let me skip forward. We know that this is the optic stalk, we know the optic vesicle, we know the surface ectoderm. Fast forward, we know that there happens to be a hyaloid vessel which comes from bottom, goes and attaches itself there, and kind of supplies the developing lens vesicle. Fast forward, we know that as the progression happens, our green friends are also coming and slowly invading without our notice. And you can see the primary lens fibers is here, the future corneal epithelium as well. Fast forward. Suddenly, without us realizing, all the endothelium forms from the cornea, all the green stuff again kind of invaginates forward. And we have the corneal endothelium, we have the corneal epithelium, we have the eyelids which are formed. The vessels are still there. Please note that the vessels are still there. And as the weeks progresses forward, our important event of the eyelids fusion happens in week 8. And then now, as you can see, the regressing of the hyaloid vasculature. The iris sphincter forms, we know that the hyaloid artery regresses. And then finally, we form the whole eyeball. So basically, this is in short what the embryology of eye is. And this brings us to the end of the lecture. I would be very happy to take any questions if there are. Thank you.
Hello, Dr. Aditya. If there are no questions, it is a very difficult topic to even understand and present, but I think you've done an excellent job out of it. One of the best presentations in recent times I've come across.
Thank you, ma'am.
Yes, and I think Aditya set the benchmark now. The rest of the batch.
Yeah. Yes,
we've
ma'am.
seen how it is done, and we expect the same level every time. All right, so you revived the seminar. The seminar that was dying a slow and painful death has been revived by you. Very well done. And keep
Thank
this
you.
up. Okay?
Thank you,
Yes.
ma'am.
Okay.